I will introduce now our next speaker, Debora Silva Pinto from the Federal University of Rio de Janeiro. Deborah is going to be talking about the characterization of St. Louis encephalitis virus, neuroinfection in central nervous system, human cell models, and IFNAR knockout mice. Um, Looking forward to hearing from you, Deborah. And again, if you have any questions, feel free to put them in the chat along with lots of support for Deborah. So off to you.
Yes. So thank you for the introduction and thank you for the opportunity as well. So I'm going to start to share my screen. Can you all see it?
Yeah, you're good.
Sure. So again, thank you for the opportunity. So as in the introduction, my name is Debora Silva Pinto. I am an undergrad research student at the Genetics and Immunology of Viral Infections Laboratory, vinculated at the Microbiology Immunology Institute of the Federal University of Rio de Janeiro. I've been supervised by Dr. and Professor Luciana Barros de Arruda, and also Dr. and Professor Charlton Vinicius Antônio Coelho. And I'm here to present a work titled Characterization of Saint Louis encephalitis virus neuroinfection in central nervous system human cell models and mice. But what is this virus? So St. Louis encephalitis virus is a retrovirus virus transmitted by Culex mosquitoes that is capable of infecting humans. Most of the infections are asymptomatic, and when symptomatic, portrays generic ones. However, the infection aggravates once the virus reaches the central nervous system, progressing the infection to a severe state, which can cause, among other symptoms, convulsions, agitation, meningitis, and the hallmark of the disease, encephalitis. At this state, the mortality rate can go up to 20%, and those who do survive may live with neurological sequelae such as cognitive impairment. As you can see here in this map, the virus has been circulating the Americas in both north and south region of the continent and been causing outbreaks on both in both sides, in both hemispheres. In Brazil, it has been detected in clinical cases and potential reservoirs, here represented by this paper from last year that detected the virus in non-human primates. However, clinically, its diagnosis is a challenge due to cross-reaction serological tests with other retrovirus and also the lack of specific symptoms in most of the infections. So usually the case reports come from either retrospective analysis from more common retrovirus outbreaks, such as this case, where a sample was collected from a dengue and Zika outbreak, or cases where the patient presents very specific neurological related symptoms, such as this case in Amazonas, where the patient presents vertigo, tremors, compromised gait, and encephalopathy, which are alterations in the structure or function of the brain. Though presenting emerging potential and being a threat to the public health, the virus is still neglected and is poorly reported in the literature. Searching for papers that regards this virus specifically, we have less than 300 results. Many of those are case reports, phylogenetic analysis, and immunological studies, and few of them address the pathogenesis of it and basic virology. So it's natural that the literature, there are some gaps regarding it, and here I brought some of those. How does the virus reach the central nervous system is yet unknown. So the viruses in general that reaches the system have to cross the blood-brain barrier, which is a barrier that separates the brain from the circulatory system. There are several ways to infect the system. An example would be the infection of the endothelium, which is the main structure of the blood-brain barrier, and the producing of infectious particles to reach the system. But for this virus is not yet established. The target cells are not yet established as well. There are a few papers regarding the characterization of the central nervous system resident cells upon SLEV infection, but it's still not clear which are the target cells. And also, if an in vivo model can be infected with SLEV with an IV inoculation that mimics the natural route you would take. So therefore, our work aims to characterize SLEV infection in resident cells of the central nervous system by evaluating susceptibility, permissiveness, and viability. Here, we're evaluating 4 specific cell types. We're looking to endothelial cells using model HB max cell line, since it composes the blood-brain barrier. astrocytes using model FHA cell line, since they're immune regulators and modulators of the blood-brain barrier, microglia using model C20 cell line, which are immune effectors of the central nervous system, and neurons using model SHSY5Y cell line, since their role in receiving and sending message, among other things, in the system. And also, we aim to establish an in vivo mouse model via systemic IV inoculation. So going to the methodology, all 4 cell types underwent the same process. All were individually cultured and inoculated with a MOE of 1 or 0.1 with SLEV and monitored to 72 hours post-infection. Cell lysate was collected up to 6 points in the kinetics for intracellular RNA quantification through RT-qPCR, thus elucidating if the cell is susceptible to the virus. Also, supernatant was collected up to 6 points of the kinetics for 3 purposes this time: for quantification of extracellular viral RNA, viral quantification by plaque assay, thus answering the permissiveness of the cell, and also viability. Here we use the release of the intracellular enzyme LDH as a parameter or cell death. So going from our results in our in vitro models, first in the endothelium, we can see that throughout the infection, the cell is capable of producing more virus, reaching its maximum in 72 hours post-infection. From 6 to 16, it actually already doubles, but it continues to grow steadily until 72 hours post-infection, reaching at its peak around 10 to the power of 6 plaque family units per milliliter. And to see if this infection is leading to cell death, we conducted a viability assay. And as is shown in the graph, the later times presented a decrease in cell viability, indicating that this cell may die from this infection, reaching at 72 hours post-infection a viability close to 70%. So HBmax is susceptible and permissive to SLEV infection, losing cell viability in later times. But why is this relevant? So as I said before, endothelium is the main component of the blood-brain barrier. And not only we have a virus that is infecting those cells and replicating in it and producing virulence to the central nervous system, but it also decreases the cell viability, which increases the BBB permeability, the blood-brain barrier permeability, facilitating the entry of SLEV into the central nervous system and putting in danger the resident cells, such as astrocytes and microglia, that are closer to the structure. So analyzing our glial cell models, we can see here that they also are permissive to this infection. We can see both that, like HBmax, their peak production was at 10 to the power of 6 perfuse per milliliter. So they are also permissive to this virus. Regarding our neuron model, the SHSY5Y cell line, we first quantified this viral intracellular viral RNA. And as we can see here, in this graph, there is a considerable increase, reaching at 72 hours post-infection, RNA equivalent to perfuse per milliliter of 10 to the power of 12. So there's a lot of viral RNA equivalent, but which means that it is a susceptible cell, but it does not mean it's permissive. We also quantified the extracellular RNA, the extracellular viral RNA, as you can see, has the same pattern and continues to grow until 72 hours post-infection. Which reaches 10 to the power of 8. But again, this by itself does not mean that the cell is permissive, but the plaque assay answers that. And when we look at this, we can see that they both have similar patterns. They both have the same pattern, and they both have similarity in the production. So we can see here that the RNA equivalent to perfuse per ml in 72 hours is 10 to the power 8, while the actual infection particles are 10 to the power 6. Also, we conducted a cell viability assay to see if this cell also dies from the infection. And as we can see here, like HBmax, it does in the later times of the kinetics. At 72 hours, the cell viability of this of the neuron model reaches around 60%, which is a considerable amount for us when analyzing cell death. And this is meaningful because excessive neuronal death can cause irreversible neurological damage. So this can be linked to the disease somehow. And going to our in vivo models, we first aim to establish a preferred mice and preferred inoculum. So we first inoculated through IV different doses of SLEV or MOC in wild type and IFNAR knockout mice, and evaluated daily for weight, clinical signs, and until their deaths. And one can see here in the weight graphs, we can see that the higher the dose starts to lose weight around 3 days post-infection. Here represented in both cases, here represented by 10 to the power of 5. Here you can see the triangles decreasing for the 3 days post-infection mark. And also here in the IFNAR knockout mice model. And those who received the lower doses, like 10 to the power 3 or 10 to the power 3 here as well, start to decrease around the 5 days post-infection mark. When looking at their survival, they both started to die at the 5 days post-infection mark. And also, as suspected, the first to die was the ones that received the highest dose of inoculum. The clinical signs between those two models seem to be pretty similar. So here I brought a representative for the both of them. Here is the wild type clinical signs throughout the infection. And we can see here a couple of information, which is expected. The mice that received the higher dose start to present the clinical signs earlier with a more aggravated symptoms. And also, which for me was interesting at least, is that all concentrations start to aggravate more around the 6 days post-infection. And lastly, that regardless of the concentration, all of the groups presented neurological related symptoms such as paralysis, seizures, and motor coordination impairment in the later phase of the infection, which induces us to believe that this virus is neuroinvasive. But we cannot affirm that just by the clinical signs. What we can do is by analyzing the viral load in the brain, which is what we did. So here we collected spleen, brain, liver, and lung in the case of the wild type to quantify the viral load. We collected spleen and liver for the connection, the highly connection of the tissue with the blood and brain because it's our main goal to establish it. And we can see that in both models, in the final stage of the disease, that's where the mice died, the viral load is almost solely in the brain in all of those animals, reaching close to 10 to the power of 6 also. So for us, it was really relevant to see that how neuroinvasive this virus is at its later stage of the infection. So in vitro, we can say that all 4 cell types models show to be permissive to the infection. And in vivo, we can say also that SLEV can affect both immunocompetent and immunodeficient mice models via IV inoculation. SLEV is neuroinvasive through IV inoculation, and the viral load is almost entirely in the brain at the final stage of the disease. We have big plans for the characterization, but here are some of our future perspectives. So in vitro, we aim to evaluate astrocytes and microglia immune cellular response upon the infection due to their immune role in the system. And also in vivo, once the preferred mice and inoculum is settled, we aim to characterize target cells in the in vivo model and evaluate the inflammatory response in the brain of an IV inoculated mice with SLEV. I want to thank you all for your attention and also my lab and collaborators. Thank you.
Amazing talk, Deborah. Thank you so much and congrats. Can I ask what types of things you're looking for in the immune response in vitro in those microglia and astrocytes? What types of measurements maybe you would take? Sorry.
Sure. So astrocytes, we haven't started yet, but with microglia, we started to look at ISG, so interference, genes that are regulated for interference. And we also have some preliminary data comparing human microglia to microglia in mice microglia, and we can have some differences. So we are first aiming to this direction to see if interferon is important to the infection, if there is an antiviral response or there is an antiviral response blockage on those cells.
Amazing. Okay, super interesting stuff. Some other questions that are coming in from the chat as well. So are you able to share what the mechanism of entry SLEV into neurons and microglia is?
the entry in the cell, I don't think they're
Yeah,
established.
how? Okay,
I don't think they're established yet in the literature how it enters the cell. So I'm
okay,
not aware how can I respond to that. But I can take a look of it
totally.
after and see if I can answer via some way of communication.
Yeah, in the chat, or maybe it's a later part of your work. So super interesting as well. The follow-up question to that is, do you know of how it migrates to the brain, maybe from the periphery at the time of infection? Or is there any established information on that?
It haven't, I think there is not established yet. What we aim to do also with the in vivo model is to sacrifice the mice in strategical days, so 2 days post-infection, 5 days post-infection, to see the viral load in the target, in what we think would be the target tissues, and how is the concentration. So again, I say that liver and spleen, they're not viral load in the final stage, but it does not mean that in all the infection, they are not there. So it's something that we're seeking to understand and how many days it takes to the virus to reach the central nervous system and how it is their pathway throughout the body. So it's also something that we aim to do in the future perspectives with the in vivo studies.
For sure. And I guess timing is probably a pretty big question of a lot of these tests that you're working with, right? Like looking out to 5, 7 days, and
Yes.
also in the in vitro test, looking out to like 72 hours. A lot of your work was showing. I was wondering also if you tested anything beyond 72 hours. Do you see the viability continuing to decrease in that time or any changes that come up?
We haven't, with these cell types, we haven't expanded to 72 to post 72 hours post-infection. What we did once was with our neural model with a lower MOI to see if the cell dies post-infection and post that time. And we see that it was not by, we see a certain death, not by LDH release, but by other type of death. So yes, it can have difference after that, but we in our lab, we focus in 72 hours post-infection, which are we think they are the crucial hours of the infection. But sure, it's something that we can take a look after, but it's not our main goal right now.
Totally, for sure. Um, and you definitely have a lot of fans in the chat. One of their questions is, how do you plan to expand on your research within Brazil, maybe in the context of policy or your next steps, or any comment on that?
So right now, I think that is because such a known virus when I read in the literature, I get really excited to what I want to try next. But first, I want to work with the astrocyte response. The molecular response already been working with another colleague of the team, so I want to take the lead in the astrocyte response. And in the vivo studies, I want to also see and through the literature if there is any indicative of other tissues that I might take a look on, something that I was not expecting, such as this primate that I mentioned, the non-primate, non-human primates. Uh, there there is an identification of this virus in a saliva, if I'm not mistaken, and the glandular saliva. So for me, it was really exciting. So it's something that I want to take a look on. And yes, I think there's a lot of ways, since there's in the beginning, there's a lot of ways we can take that, but it's still in the beginning, so we have to take a step at a time.
Absolutely. And what a great first step you've taken. And you'll have to come back next year and keep us updated on the progress of all your work. So thank you so much for
Thank you.
sharing all of this, Deborah. It's so interesting, and congrats again. You really did amazing today. So well done.
Thank you.
If there are any other questions for Deborah, please feel free to put them in the chat and we will leave those for you to address if that's okay.
Okay. Yeah, in a couple of minutes I have to leave. But you can
Totally.
share my email address if for other questions or whatever. Thank you.
Anyone's in the chat has those in the next few minutes. So Deborah's got to run. So thank you so much and congrats again.